thank you very much for the invitation and for the introduction. So I'm going to be talking about the studies that we have been doing in the laboratory for the last several years, trying to identify what are the features of peptide fragments that are presented in the autoimmune model of NOD diabetes, which is very similar to that is occurring in the human disease. How I've divided my talk is by first giving you some general features of antigen presentation, because I like to bring that into the context of what we are studying. Parenthetically, there are many facets of autoimmunity that you can study, of course. The one that we are concentrating on, our niche, is to try and identify what are the immunogenic peptides and their particular features. That's where I think we can make a contribution. The second issue that we're going to consider then is the autoimmune diabetes. I'll talk in general about the features of the class 2 molecules that are very unique, by the way. And then I'll continue with these items of using insulin as a major autoantigen and analyze how it is actually being recognized in the islets of Langerhans. So this is the issue that we are trying to identify: what kinds of peptides from autologous proteins are being processed and presented by the MAC molecules. We are entirely focused on class 2 MAC molecules, which in the diabetes system is the major component that is responsible for the autoimmune diathesis. And what we want to know is that whether these self-peptides have any particular features. And if so, in the target organ, how are these self-peptides then presented? So we all accept that autoreactive T cells that are giving autoimmunity, and I'm focusing entirely here on CD4 T cell autoimmunity, have escaped. They're in the periphery and actually are responsible for the process. So the issue is, why do they escape clonal deletion? Why aren't they purged in the thymus gland and eliminated? Why is that process not exactly 100% effective? And if so, does this have to do with the particular features of the peptides that are being presented? So these are some of the issues that you can then discuss. You can think about the autologous peptide MAC complex not being expressed in thymic APC. I don't think that's the case. There's more than enough evidence to substantiate the fact that there are most tissue-specific antigens, or many of them, are expressed in the thymus gland, and there's the air issue, the air control of this. The thymus also is an exquisite organ in trapping self-proteins and actually presenting proteins that are in the circulation. And many self-proteins actually have a certain cycle of circulation in blood. So I don't think number one is a feasible one right now. The issue then comes in, what about the peptides that are really then driving the process? And you can make a point that many of the peptides are very weak binding to forming peptide MAC complexes. And that has been an issue that has been discussed in the issue of some of the peptides that induce, for example, autoimmune encephalitis. The basic protein peptides are extremely weak binders, yet they're driving the process. And as I will show you with insulin, that is also the case. There's very weak interaction between them and the MAC molecule. So you can make a point then, because they're weak, they are escaping the thymus negative selection process. Now there's another issue, which is the one that I like to focus on with you today, is that the peptide MAC complexes that are presented in the tissues by the antigen-presenting cells of the tissues are different from those that are presented in the thymus. And therefore, you are selecting a number of T cells that are seeing a somewhat modified epitope in the periphery. And you can say that they are different because they have post-translational modifications, or because they have unique conformations of their peptide MAC. A given peptide can form very stable complexes, but it can also form rather unstable complexes. And you can make a point that maybe in the periphery some unstable peptides are presented. Or you may have unique registers of a particular peptide in the way in which they bind to the MAC in the thymus might be a bit different from that in the peripheral tissue. I think this is the case. So what I have right now is I have 3 or 4 slides in which I'm going to go to some basic aspects of antigen presentation. And through that, I'm going to bring into you what are some of the features that are important then in the peripheral presentation of self-antigens. So this is a very simple scheme in how antigen presentation can go in the class II system. You have 2 vesicular compartments, early or late. The early is an endosomal compartment that recycles, contains class II molecule diagram here as this little bar in here, like this. Now, when you have actually a protein, this is the classical pathway of presentation. The protein is not open up into the very early late compartment because the reductive activity is not in the early recycling compartment, but in the late compartment that receives class II from the endoplasmic reticulum and Golgi, and where there is enzymatic proteolytic activity. So a protein like the ones we've been studying has to be transported to this late vesicular compartment. In the late vesicular compartment, the protein is open and reduced and fragmented. And there it binds through a process which is very complex in a way to the class II molecules, and then it's transported to plasma membrane. In this late compartment, you have a number of auxiliary proteins that help in the assembly of the protein. The MAC molecule is coming with the invariant chain, and there's a small peptide that gets cleaved, which is called CLIP, that is released, and there's a chaperoning molecule, which is H2DM or HLA-DM, which edits this complex that is formed in here. This is an important protein. It's an important protein because it interacts with the class II molecule as it binds peptide and changes that interaction. It favors the good stable peptides. It actually is unfavorably from the unstable peptide. It's a peptide editor at the end. So the peptides that are formed in these compartments are relative good peptides that are transported to plasma membrane. Well, if you have an APC that is actually activated by interferons or TNF and so forth, this APC then can actually produce a number of molecules that actually can oxidize these peptides. So you might have actually peptides in here through this process, if the APC is activated, that actually have a number of oxidative changes. And this will create neoantigens in the periphery in a situation where these neoantigens weren't produced when this protein was actually expressed in the thymus, for example, in the context of autoimmunity. Now, there's a second pathway in the class II system, which is the pathway where autophagy molecules actually coalesce with these late vesicles and form an autophagosome. And then products of the autophagy molecules, which are cytosolic proteins and proteins from other organelles, are brought into the class II system. Well, in the autophagosome, actually there's a number of new enzymatic activities, and one of them is to actually citrulinate peptides, change the arginine to citrulline, and in fact generate these. But the point is, from the cytosol, you can have a number of proteins presented in the class II. If you isolate all of these peptides from a class II peptidome, about a third of them come from cytosol. So this is a very effective pathway that is taking place. But the point is that through this pathway, you can generate these unique peptides. And you know that in rheumatoid arthritis, for example, there is a high incidence of antibodies to citrullinated proteins, which we are linking to this autophagy process, which can be induced in the cell or be constitutive. And then there's the third pathway, which is the one that we've been interested in which actually what happens is that exogenous peptides, not the protein, but exogenous peptides from this protein, are actually presented to the cell. These peptides do not get to this compartment. They are actually bound to class II molecules through peptide exchange and generate a number of peptide MAC complex. The concept here, which is important, is that in this compartment, you do not have the editor molecules, or if you have them, you have in very low level. So at the end, you generate here a number of peptide MAC complexes that are unstable, which from the protein were actually eliminated here by H2DM. So a peptide from this protein, which is exactly the same as the peptide generated from catabolism, may generate epitopes in here that are different from these because of the fact that in here the bad conformers that were formed were edited off. In here, they were not. And this is a fascinating result, which you will see taking place in the insulin molecule as I go into diabetes. So at the end, for example, in the protein lysozyme, which we've studied for years, a small little protein of 12,000 molecular weight, this is a major epitope in here, which has been very well studied and dissected in a number of ways. This is a side view as it's bound to the class II molecule. There's a tyrosine in here. You find many T cells that, if the macrophage gets, the APC gets activated, where this tyrosine is nitrated, giving rise to nitrotyrosine. Well, you have then T cells that are specific to nitrotyrosine and that do not see the wild-type peptide. They're highly neoantigen specific. And this happens too with the citrulinated one. If this arginine here is actually citrulinated, you will give rise to very highly specific T cell to the citrulinated epitope, and so forth. This tryptophan can be oxidized. And it's very beautiful. You can get your T cell cloned and see the high degree of specificity for these kinds of changes. Now, this is the issue that about the conformation that I was telling you about. And we've been recognizing this in the last several years, and we think it's important, particularly in autoimmunity. There's a conventional type of T cell, which is this one in here, that we all examine and use in our routine, which, for example, against the protein lysozyme, you identify the epitope, you take T cells against that, you react it to the peptide, and it reacts beautifully, you react it to the protein, and it also reacts. These are the conventional T cells. But these are the ones that we were interested in, and they are a large population of them, in which actually they react to peptide very well, but they don't react to the protein, and we call them type B. And the reason for it is what I was telling you about, the issue where the peptide was generated, the peptide MHC in the cell. So to recapitulate, what happens then is if you have a standard antigen, it goes to the late vesicular compartment. It only has stable peptide MHC complexes. Those are the ones that give you rise to these T cells. If, on the other hand, you have a situation where the protein antigen got into another cell, the cell spits out the peptides enter the presenting cell, but they do not go to the editing compartment. They're in the recycling compartment. So a number of unstable complexes are formed that now you have T cells that only see them here, while here they were eliminated. These are the ones that you saw here. And you'll see how this occurs in the type 1 diabetes model. So a single peptide MHC has conformational flexibility, giving rise to some conformations that are stable and some unstable. The proteins that are processed in the late vesicles will generate the stable, or type A that we called, while the unstable ones are removed by the H2DM molecules. With peptide, you don't have DM in these compartments, so you actually keep the unstable conformers on the periphery. Why is this important? Because in the situation in the thymus, and we have proven this with foreign proteins, or making transgenic mice to these proteins, what you have is a situation where the thymic APCs are only processing the proteins in the late vesicle and only presenting the stable one. These are going to be eliminated. They're not going to peripheralize. While these ones over here, which were the unstable ones, have peripheralized and will get activated if by any chance you have this kind of situation taking place. And what I'm going to try and sell it to you, is that this is exactly what occurs in the islets of Langerhans. Peptides are released from the beta cell, the intra-islet APC captures it and presents conformers in here that are very distinct and have escape negative selection. So now let me jump now to the studies on the NOD and try and bring this concept into this autoimmune situation. To me, one of the most important papers in autoimmunity is this one that John Todd Bell and Hugh McDevitt actually reported in 1987. And they correlated a class II MHC genotype with its susceptibility to type 1. It's the most important gene giving you susceptibility to type 1 diabetes. Analysis of DNA sequences from diabetics indicates that the alleles of HLA-DQ beta determine both disease susceptibility and resistance, and that the structure of the DQ molecule, in particular residue 57 of the beta chain, specifies the autoimmune response. This is a fascinating conclusion and a true one, I believe. and a very important one to then keep in mind. Subsequent to that, you actually identified that the NOD mouse strain that was being used in type 1 diabetes had exactly the same situation, the same biochemical features. It's a powerful result comparing the two. And I have to tell you that aside from the lupus mice, the NOD is one of the unique strains where you get diabetes spontaneously. You don't have to induce it. And this is one of the good things to study this disease. So in very brief, all MAC molecules have an aspartic acid at the beta chain position 57 that establishes a salt bridge, an ion pair with an arginine on the other side, and closes the class II molecule, what is called the P9 site, where the residue at P9 or a peptide actually binds. Okay, and that closes, in a way, the class II MAC groove. All MAC molecules have it, except the human DQ8 and DQ2 and the mouse IAG7, which they do not have aspartic acid. That leaves, this is a structural feature study that was done by David Fremont in my lab, subsequently looked at did the structure of the also of the G7, and subsequent Wiley did the structure of the human HLA DQ8, and they have similar, or if not identical features, particularly at this P9 residue. What it leaves is this arginine on the other side is left completely unpaired. And therefore, because of that feature, and this P9 area right here, you select very, very unique peptides in here. And what we've done in these studies was actually isolate class II molecules from antigen-presenting cell, forget about the islets or Langerhans, culture large amounts of them, and then by mass spectrometry analyze them and get what is the class II peptidome of the AG7 or the DQ8 molecule. And the interesting thing about it, which is very unique in all class II molecules, is the high, very high incidence of peptides that have a particular sequence. And that particular sequence is having aspartic acid or glutamic acid at the P9 at the end of the peptide, at the carboxy end of the peptide. But it's not only that. They have 2 or 3 acidic residues at their C terminus. Okay? It's a very, very interesting feature of it. This is an examination by mass spec, a summary of it, in which only we're projecting to you what are the peptides that are found at the end of the peptide, what are the residues at the end of the peptide. And most of them, as you can see by this bar here, are the acidic peptides. If you do the same study and you make a new construct now, where actually the G7 molecule you change to wild type, so you reconstitute it with aspartic acid, now you change this completely now to a different peptidome that you've isolated, and you don't get diabetes. So that particular amino acid is the go, no-go decision in diabetes. And that makes it a very strong issue to study in here. This is actually a combination of many experiments where these are the 9 residues that are bound in the groove, and this is plotted in a different way, the same situation that I have in here. So in essence, therefore, the class II molecules that give you the propensity to diabetes have a very unique peptidome that they actually identify. And this is one example of one protein isolated from class II molecules, and you can see what I'm talking about. Here is the core residue, which we know is bound to the MAC groove. Like all class II peptides, they have flanks, flanking peptides of different length because the cut as you process is irregular. And you can see here in this case 3 acidic amino acids at the carboxy end. So you find this very frequently, as I said. You can find EXD or DEXE and all these variations on that. By a number of binding studies, we know that this is the residue that is actually the P9 residue. So in essence, by a number of binding issues, you can show cooperativity of these acidic acids on the carboxy end, okay, favoring the interaction with the peptides. Now let me move now then to the insulin situation, and where most of the talk now will be. First of all, what is fascinating is that T cells to insulin are abundant in islets of NOD mice. And this has been extremely well done by the group in Denver that works on diabetes, George Eisenbarth and a number of his collaborators, who were the first ones to call attention to this. And they should have a lot of credit for these observations where no one believed that insulin could be the autoantigen. They showed them they identified some T cells to insulin, and they showed that they were actually biologically active. Subsequent to that, others have actually placed insulin at high expressed in the thymus and showed actually an effect by increasing purging of the insulin repertoire in the incidence of diabetes. So this is an important observation, and this is a major T cell component, it seems, may not be the only one in the autoimmune process of the NOD mouse, and most likely in the human. In the mouse, you have two insulin genes, insulin 1 and 2. In the human, you only have one. The human gene is very similar to the mouse gene. There's only 2 or 3 amino acid differences. The majority of the T cells against insulin are recognizing an epitope that is in the B chain of insulin from residues 9 to 23. You could say that the majority are recognizing that stretch. That stretch is the dominant one. But the fascinating thing is that if you actually take the insulin protein and take that peptide and do a number of kinetic studies and binding studies, you find that these peptides in here bind to insulin extremely low affinity, about 1 or 2 micromolar. That's pretty low. The circulation of insulin in blood is 1 or 2 nanomolar. And yet, this very small protein seems to be the driver of this situation here. And I found that shocking, that you could have a protein that is in very low amounts, that is very tiny. You have a foreign protein that you try to immunize, and it's a foreign protein of this size. You get a lousy response. Foreign protein, the best responses are to larger proteins that are more heterogeneous. Yet, this is a driver, seems to be, of the autoimmune response, which to me was very surprising. It can bind here into register, by the way, with this glutamic acid at P9 or this glycine in here. So this is the question that we were asking ourselves when we did the biochemistry of the insulin molecule. How can we generate this? What we did, and this was done by Jim Mohan, I'm a graduate student in our laboratory, we went to the islets of Langerhans of early diabetic mice or to the peripancreatic node, recovered a large number of T cells, made a very large bank of T cell hybridomas, and looked for these insulin-specific T cells. And to our surprise, we found 2 sets of insulin-specific T cells. Okay? The lower number of them, the minor group of them, had these kinds of features. In that they reacted with the insulin protein very nicely, although at low affinity here. Usually proteins are on the lower side of the scale in here, or with the insulin peptide that I told you about, this segment in here. But these were the minority of them. Okay? And these are what we were terming the conventional, the type A. But the other point was that the majority of them was the other way around. They saw the peptide beautifully, but they didn't see the insulin protein at all. And these were the majority of the cells that you find spontaneously. They are not induced. They are that what you are actually pulling out of those early islets or the lymph node. And these are what we were calling the type B. Of 351 clones that were done in these first experiments, 98% were these guys over here. Okay. And Jim and us, of course, were fascinated by this particular observation. What did it mean to have this particular set of peptides in here? There is one trick that was done that actually gives you the black-white situation. You simply construct an antigen-presenting cell where you have the 9 to 23 sequence anchored covalently to the class II molecule, a trick that was first done by Kaplan and Marag years ago. So you have an antigen-presenting cell that now has a stable, locked-in complex, and they're all presenting only that complex. That particular APC line only presents to the set of type A. The set of type B don't see it at all. So even all the presenting cell that you're using here, all the PMACs are of 9 to 23. That's only locked in one conformer that only sees the minor set of T cells. This one's in here, and not the majority of the T cells in here. So the issue is we can make a lifetime working with one strange T cells. These are not strange T cells. They were the most abundant. And to prove that they were actually biologically active, what you do is you clone them, and you actually then transfer them to an NOD mouse that has no lymphocytes, an NOD RAC1 knockout or an NOD skid. And all of them produce inflammation in the eyelids, and many of them actually cause diabetes with severe inflammation and so forth. In other words, they're as diabetogenic as any of the classical T cells that had been found before, and they're very actively diabetogenic. So the issue for us then comes in if these T cells, this is the repertoire that escapes natural negative selection, and it's in the periphery. How can you prove it? Well, one very easy way to prove it is get your mouse before it gets diabetes, or get the NOD males where the incidence of diabetes is very low, and immunize. Immunize with insulin, immunize with your peptide. Or take the black 6 mouse that expresses the AG7 molecule and immunize insulin and with the peptide. And these are the results of it. So this is an ELISpot result. You have immunized with insulin now or with the peptide. So you've immunized with the peptide that you found in the spontaneous T cells in the eyelids. And then you develop your ELISpot by taking your CD4 T cells 7 days after immunization and mix them with APC that have no antigen or have the peptide or have insulin. So when you immunize with insulin, you don't get anything. It's like if you're tolerant to insulin. And in fact, those are the minority of the T cells you found in the eyelid. There are very few here, if any. So when you immunize with insulin, this is background, that's your result. But immunize with a peptide now. Now you get it. Very nice response, 100 spots per 10 to the 6, which is a very good response, as well as you get when you immunize with a protein in Freund's adjuvant. So now, these T cells, as you showed spontaneously, were there. You can immunize them and recover them. These T cells are not seeing insulin because the same CD4 that you develop against insulin are here. That's why I'm telling you about. These are the special T cells, peptide specific, insulin protein negative. Okay? So by finding them spontaneously in the eyelids and by then actually immunizing, you can see that this is the dominant repertoire that has escaped thymic negative selection, and it's there spontaneously or you can induce it. By the way, if you do this assay and in the assay you put antibodies to the class 2 molecules, you drop it from here to here, but an irrelevant antibody doesn't do it. So in essence, the issue then is clearly that this seems to be the repertoire, this type B T cell that has escaped then negative selection from the thymus. So the kind of scenario that then we're working on that we think is happening is diagrammed in here. The thymic epithelium, the thymic APC system is presenting insulin protein and is eliminating your type A conventional high affinity T cells. A few escape, which are the ones that you find in the periphery, the few that we were finding in the eyelids. But the majority of the cells that are reactive to the insulin peptide is not presented in here. They escape, and that's why the bar graph is thicker. Those are the ones that are in the periphery. Okay. So then we have these conclusions that the majority recognize only the peptides and do not recognize the peptide from insulin processing. These are the unstable peptides that I was telling you about. Both subsets are recruited during the spontaneous infiltration, but this is the predominant subset. We have done experiments, which I am not presenting to you, in which we use mice that were created by the Eisenbarth group that are hormonally active. They knocked out the insulin 1 and 2 gene. They replaced with a hormonally active insulin 2 gene, but in which the actual epitope had been changed, so there was no insulin autoreactivity at all. In that situation, then you have these T cells escape, and you can actually study them and compare them to the others. So the issue then for us, if we're going to activate these in the periphery, as they are, okay, where are the peptides coming from that turns them on? Weak affinity binding peptide. And what I'm trying to convince you now in the next series of slides are these peptides are coming and are being presented in the actual islet or Langerhans. I don't want that. What are you talking about? Okay, so that's the question. Where are the insulin peptides that activate this set of T cells now being generated? And this is where you're finding our studies going at this moment. So let me then digress to tell you about some issues, structural features of the islets or Langerhans. At the end, the message in here is that the tissue APC is a major component by the way in which it handles proteins, the autologous proteins, in directing the autoimmune reaction. So the islets or Langerhans has interesting circulatory features. First of all, the circulation is not like the renal glomerulus, very fast. It's very slow and intermittent. Not all capillaries of the islets are open at the same time. Secondly, very importantly, it has a fenestrated endothelium. It has holes in the endothelium. And thirdly, normally, the islets of everybody, including all of yours, okay, contain a series of antigen-presenting cells normally residing there in the islets. Okay. These anatomical features of having APC within islets normally, and the fact that you have, you will see that you'll have also leads to the presentation of peptides from secretory granules, an issue that I'm going to discuss, and will eventually influence the presentation and the specific localization. Here's a study that was done by Boris Calderon in our lab, in which he isolated pure islets from the mouse. Okay, these are not histological pictures. I don't think this is very easy to do or at all possible in histology. You have to isolate the little ball of cells with 1,000 cells and then examine them. And you can take those little organ cultures and actually bring in fluorescent reagents and study the cells that are within. So if you take an islet like this and you stain for PCAM as an endothelium marker, you can see the vessels as they cross the islets in this immunofluorescence. But if you look at now staining for APC markers, like in this case 11C, you find this network of APC. This is a normal islet from a non-diabetic mouse. Okay, this is a normal islet, and you can see them wrapped around the vessels, very tight associations around the vessels of this APC network. They're throwing dendrites, okay, and you see this very, very close association of them that you can pick up if you do EM and are patient enough to section the way through. You can see actually instances where you see your vessels, red cells, the beta cell with the granules, and these APC plastered then next to the vessel wall. And in fact, this is a very pretty picture where we happen to get, by some chance, the dendrite extending along the vessel wall. Here's the capillary. Here's a leukocyte that was throwing a pseudopod in here. And this is the fenestrated endothelium that I'm talking to you about. Okay? So conclusion, yes, there's an APC network normal in the islet. You can study them. They have 11C, 11B. I think they are monocyte derived. They stay in islets for long periods of time. They're very, very long-lived, an issue that we are now considering. And it has a certain special biology. These are not reconstituted by pure dendritic cells. So what we've done then is go back to the insulin situation, knowing this. And we made a monoclonal antibody to that insulin peptide that we know where the immunogen was. And the monoclonal antibody only reacts with the peptide, does not react with the insulin molecule. It's a peptide-specific monoclonal, like many that you can do, which doesn't see the peptide in the restraints of the normal conformation of the protein. So when you do this, you find these following results. First of all, many of the dendritic cells that you're seeing that are situated between the beta cells capture granules. And you can see one beautiful example in here. This is a beta cell granule. Okay. We consider about 8 to 10 granules per DC that are within the islets of Langerhans normally. A dogma that has come up in diabetes is that your antigen-presenting cell have to see the antigen if the beta cell is dying. This is not the case. All the cells here, even from NOD, RAGs, and so forth, are presenting peptides, okay, and are capturing secretory granules in a constitutive process. As the beta cell degranulates, Okay, some of these granules are taken up by the APC. Okay. And each granule has 10 to the 6 molecules of insulin. So start putting in the results of a protein that gives peptides at a very low and weak affinity and yet brings in T cells. Well, you got a situation in here where you have a large number of granules being captured, of which contains an incredible amount of protein, or the target. But if you now take your monoclonal antibody, this is the whole islet, these are the DC, and you can see that the beta cell contains a lot of the granules in the beta cell have this particular peptide. So they have insulin, C-peptide, but they also have this particular fragment from insulin degradation. We've isolated these granules, done extensive mass spectrometry analysis, and indeed they have insulin C-peptide, and they contain that particular peptide. So, and this is a combined micrograph in here where you can see your islets, your DCs in red, and you can see your granules stained with these monoclonal antibodies. And in this example, I think there are about 8 of these granules inside the dendritic cell. So you can confirm this by not only your micrograph in here, by electron micrography, but by confocal microscopy too, and chemically. So in essence, these peptides are coming in 2 ways, we think. One is because the insulin granule in itself, okay, is degrading part of the insulin molecule and contains the peptides. And secondly, because when insulin gets into the cell, it also releases some of these peptides. So to prove this, what you have to do is get your islets from an NOD skid mouse that has no lymphocyte, get your islets, isolate them, release your APCs, and then test them against your clones that you have isolated. And if you do that, then it's only these APC from the islets that fire your type A or your type B. So they are loaded with peptide MHC complexes. So APCs from the spleen and the lymph node, we've seen no presentation to type A nor to type B, but if you isolate them from the islets, they are loaded. And that's where we believe the action then comes in. It is most likely that some of these intra-islet APCs move to the draining lymph node to further sensitization. The draining lymph node is important. How this takes place, we do not know. Islets of Langerhans have no lymphatics. So the movement from the islets, these APCs, into the draining node is something that still has to be. It's a pathway that likely exists, but we don't know how it actually operates. You can do this completely ex vivo by taking beta cells, culturing with APCs, and then coming in with your T cells, and they'll fire. And if you look at your APC, you see the capture of the granules. You see 1, 2, 3, 4, 5, 6 that have been captured by the APC. What the granule component is that it allows to be actually captured by the APC, I do not know. The question can be asked, why should islets of Langerhans have intra-islet APCs that actually could harm you if you have the other genetic components that can lead you to autoimmunity? And the answer is clear. Take islets of Langerhans don't have APC. And the way to do it is if you actually take some mutant mice like the one that have a mutation in the CSF1 or MCSF, which do not generate a certain population of APC, those islets do not have APCs. Okay? So that's what I'm telling you about. These islet APC are not the classical dendritic cells. They are derived from monocyte and they're MCSF dependent. Well, those islets from those mice are actually tiny. They are considerably have about 1/3 to 1/2 the mass of the normal islet. Okay? So I think these islet APCs, in ways that we do not know at present, and we're studying that, actually generate, have some kind of a trophic function, either through vascularization or through the health of the beta cell. If these mice, you do a glucose tolerance test, they are abnormal. You would classify them as pre-diabetics. So the intra-islet DC has a function in the sustenance of the biology of the islet itself. That's why they're there. An issue that has been found in other tissues too, by the way, not uniquely in the eyelid of Langerhans. So that's what we believe is their normal physiological process, which in the case now of having a mouse that has all the genetic component is a central cell then in inducing the diabetic process. This is fascinating, these intra-eyelid DC, to continue the discussion on them, because these intra-eyelid D3s throw dendrites into the blood lumen, into the vessel lumen. This was done by Boris Calderon in our lab, and you can see this confocal microscopy where you see the DC in green plastered to the cell, to the vessel wall in red, and you can see the little thin dendrites coming into the lumen of the vessel. This is en face. Okay, you can see them right here. So whether these penetration of these dendrites into the lumen is instrumental in bringing T cell is something that you can let your imagination go, whether or not they are central in that part of a process. But that this is the case, this is actually a very nice experiment where you take 0.5 microns coated beads with antibodies to the class 2 molecules. Okay. And then you inject them, and immediately, within a few minutes, isolate your islets or Langerhans. And you can find the beads in some of the capillaries, as you find them here. Here is the blood vessel stained for PCAM. Here's your DC, and here's the little bead that you injected, plastered right here in the site of contact. That is totally specific. You can do the experiment very nicely. By putting antibodies to one class 2 molecule or the other, you can see the specificity of this location. So what I'm getting at is that these intra-eyelid APCs are not only supplying the antigen, but they're also instrumental in actually bringing the cells perhaps through this kind of a process that you see here. In the last 3 or 4 slides, I'm going to actually move fast to just present you where we are right now, which is trying to track how these cells then can bring in these T cells. And you can do this in 2 ways. You can actually take your type B T cells. We've made T cell receptor transgenic to the Bs and to the As of insulin. You can inject them. You can follow them in the cell, in the APCs. You can do it with other T cell clones that have been isolated by others in the field. And you can do cell transfer and see the very fast localization of these T cells into islets of Langerhans, and then see what happens in this localization. You can also actually go to the very early pre-diabetic NOD mouse and look and see if there are T cells in there, which you find early in diabetes. Weeks before the whole inflammation takes place, you already start finding some of these T cells within eyelids. And here's a beautiful picture that Boris made of an entire eyelid of Langerhans of early diabetes. This is an entire eyelid. The vessel walls are seen in PCAM, in blue, stained for PCAM. The DCs are in green. Here's the typical DC that I've been telling you about, plastered to the vessel wall. But then outside the vessels, you find a number of T cells already coming in. And you can find this again spontaneously, or you can inject them and localize them. You can see them, they're out, and they're in contact with these intra-eyelid APCs. You can actually see the synapse in here between the 2 cell contacts. You can see one very beautiful one where you have 2, looks like 1 or 2 APCs and 1, 2, 3, or 4 APC plastered to it, and you can see the synapse in here. You can actually then, by cell transfer, do 2-photon microscopy of these cells and look at the time of contact. And the time of contact is as high as you see T cell interactions with DCs in the lymph node. So there is a specific entry, we think, conditioned to these particular APCs that have captured the antigens from diabetes right there. And that, in fact, that may be the ensuing event that starts the whole complicated process of diabetes. And to summarize what are experiments that are taking place right now in just one slide, as soon as these T cells are coming in, the whole eyelid biology changes. Again, directed by this very first encounter that we see here, and by the selection of these specific T cells that are peptide specific. And the changes are seen and summarized in this next two slides. And that'll be the end of my talk. So an islet of Langerhans has some ICAM, and ICAM, the adhesion molecule, seems to be important in this localization that I was telling you about. But the islet of Langerhans doesn't have any VCAM, for example. It's totally blank. As T cell starts coming in, and you can do this in cell transfers, you can see here an islet, and you can see a bunch of T cells in here, an islet with less amount of T cells, which are in red. But as soon as these T cells come in, within 24, 48 hours, the whole islet changes its biology. And you have a number of new genes now being expressed. And not only do you see that, you see more ICAM. You can see it here now compared to the control. Moreover, if you actually disperse this islet and isolate the beta cells, they have ICAM on the surface, which normally they don't, which could make them more receptive, perhaps, to attack by CD8s. But now VCAM appears in the islets, which was never there before. Okay? So they're very rapid changes, and the islet then becomes receptive to other cells coming in, where they're regulatory cells for a period of time, where they're inflammatory cells, are basic fundamental issues that have to be tackled. But the very early clean islet that receives a diabetogenic T cell immediately reacts with this kind of changes. And if you do actually microarray analysis, you see a number of upregulated genes that appear very rapidly in these islets. This was done by Javier Carrero in our laboratory. He, Jim Mohan, and Boris Calderon have been responsible for all these studies that I've been telling you about. And this is a diagram in here, one of these type of charts, where you see the large expression of new genes, most of them interferon signature, okay, in the islets as soon as the T cells come in. And actually, many of these are expressed in beta cell themselves, not only in the infiltrating leukocyte, but in the beta cell. There's about 360 new genes upregulated. They show an interferon signature, and they're expressed subsequent to the localization of your T cells into the islet. So this is the take-home message. The islet of Langerhans contains a number of antigen-presenting cells. These presenting cells are presenting products of the secretory granules. Among the products of the secretory granules are peptides that actually give rise to a very selected population of autoreactive T cells that have escaped negative selection. And these are the type B that we refer to. These islet of Langerhans, these intra-islet APC may actually be instrumental through this kind of intravessel dendritic cell in bringing T cells. There is no question that they're instrumental in the first migration of T cells into islet. I believe this may be a common features of endocrine organs, where you have secretory granules, where you have products of digestion of the hormone plus the hormone, and which you have a network of disease along the endocrine cells. I believe it happens in thyroiditis, to bring to you one of the your favorite autoimmune reactions. The thyroid contains a network of dendritic cells. Dolores Araquemada in Barcelona actually has studied these very extensively. They are normal in normal islets of humans around the thyroid follicle. I have seen some evidence from others in which T cells to thyroglobulin have the same kind of features that the ones that I'm telling you about, react with peptides, do not react with the protein. In other words, reacting with the secretory, with the products of digestion of the secretory granule. So I think this is an issue that may be common to endocrine autoimmunity, where you always have this kind of intra-organ APC capturing granules. And this is the scenario that you have for the late phase, where you have the dendritic cells capturing, and as soon as you have specific entry, you have an amplification. The islet now becomes receptive to other cells and to modulation of the inflammation. So in a nutshell, I've put you with a lot of data that has come up in the last 4 or 5 years, but I wanted just to put you up to date into what are some of these main issues, making this a central cell in the tissue autoimmunity. Of course, this is a very complicated chronic reaction with many players coming into action, but I think that this particular chemistry of these antigens and these phenomena of presentation in the periphery are major components in driving this process. Thank you very much. Not that we can tell. In other words, we've done the same studies with boys' APCs and with the APCs from the boy mouse and the girl mouse, the NOD skid, and insofar as loading those particular that is not the issue. There's a lot of factors modulating this, of course. Okay, this is the substrate one I'm telling you about. But as far as we know, there is no difference. That's a good point, but we can't find it.
This is a fascinating story, but similar to, it sounds like this would be a common mechanism that you would have a bunch of peptides presented or available for binding to the MC class 2, and that way it seems like the susceptibility relies only on the susceptibility to diabetes, maybe just dependent on whether you have the right MAC or not, but we know that is much more complicated.
Of course. Yeah.
So what else would be unique in NOD mice, for example, which would, is there a mechanism in NOD mice that would generate much higher concentration of these peptides or release them or anything?
Well, you have the biochemical features that I talked to you about. That's number one, okay? Other alleles bind very poorly, okay, to this one. So that is one feature. But again, this is the substrate. There's now a great deal of modification of this. It's the issue that identical twins in diabetes, they have concordance only in 30%. So there's a number of other influences that allow those T cells to get to an activated stage, or that goes into further activation, or that changes or modulates this basic first interaction that I'm talking about. And that is crucial in the studies of autoreactivity in general, how this basic interaction then is modulated further to actually get to the effector reaction. If you look at, it's fascinating, if you look at islets very early in the mouse, address your point. Mouse doesn't get diabetes until about 22, 24 weeks of age. If you look at the islets at 4 to 6 weeks of age, you find that picture was an islet of Langerhans from a 6-week-old mouse. There is no other site where lymphocytes are except within these islets, and you already had those T cells coming in. We have to pull them out and see their specificity, by the way. We don't know that. Okay? But as soon as you then keep on tracking those islets, okay, 2 or 3 weeks later, you have a new flush of cells coming in. You still don't have derangement of beta cells. Now you have some B cells coming in, some CD8, some FOXP3 CD4 T cells coming in, some NK. There's a whole plethora of other cells coming in. You still don't have any problem in here, okay, until much later, at which time then whole thing changes completely. This second phase, I think there's a lot of regulation taking place in here that is actually cooling down the reaction until something happens and the whole process goes further. So I think that what I'm showing you is the substrate as an example of one. Okay, the whole process is much more complex than this as it follows through.
What is it within the granules that breaks down and alters the insulin? And is that present, and does that take place in normal as well as NOD animals?
Yeah, it does. But let me tell you one thing. I don't know the first part of your question. We think, from what we've done, that there are two kinds of secretory granules. The one that has insulin and C-peptide in equimolar concentrations, but that there are other granules too that are very rich on this particular peptide. So it looks like in the normal beta cell handling and production of the mature insulin protein, the process is more complex than just having a single secretory granule with the final product, that there are others that are evading or going perhaps to lysosomes and coming out, where this are very rich in this particular peptide. So in essence, when you can do the combined histochemistry anti-insulin, anti-secretory granule monoclonals that I showed you about, you find many granules that are secretory peptide specific, no insulin. Okay? And others that are entirely insulin without the secretory, with a little peptide. So I think there is heterogeneity in this catabolism that the end product is a lot of these secretory these peptides. But the evidence is that there's no special issue here with NOD or non-NOD strains. Okay? That is taking place normally in everything. David.
How does this relate, you think, to the concept of epitope spreading? So you talked about how antigens might be broken down to sort of another situation. You look in the brain, like myelin basic protein TLP, where there are grams of protein there with tissue destruction, peptides being generated. You take myelin basic protein, for example, or common MVP, put it in syrup, you start generating epitopes which are directly presented by T cells. How does that, what you're finding, IP T cells are going to epitope spreading, and what happens with tissue?
Yeah, that's a great question. I wish I knew it all. Here's my bias, that there is a first wave of T cell that is the driver, that this may be the drivers, okay? But subsequent to that, you are having a wide diversification of the autoimmune response. That probably as you set in then certain steps, charging or inflammation, now you're getting in a much more epitope spreading. And indeed, it is true. There is epitope spreading in here. You find other specificities as it goes in. What has been very hard to do experimentally, as you can imagine, is to time this, that you have 10 autoantigens. When is this first one coming and the other? That's a tough one to do in a good quantitative way. So, but I agree. I think there is there's more than one. Although I think our thought is to actually hammer on what we may think are the first specific T cells that come in, which could be these. No.
Yeah, just to comment a little bit on two. First of all, I'm struck by the parallel synergism, perhaps, between your unusual, unconventional T cells and the idea of Eli Surkar and the Mudball of cryptic antigen. And is this the T cell equivalent of a cryptic antigen, one which is normally not well presented in the thymus, but can be presented in the
My definition is so far. My great discrepancy, with all respect to Eli, is that in his definition, cryptic antigen was unprocessed proteins, peptide. Okay, and my big debate with him was that these are peptides that are there because chemically we've isolated them. They're outside. We've isolated them chemically. They're not cryptic. They're not hidden. It has to be another explanation for that phenomenon, which the phenomenon is true. Okay. But the explanation of it is there's a big difference from saying the peptide is not processed to saying, yeah, the peptide is processed, but it has a different structure from the peptide that was processed from protein. That is where the issue came.
Follow on is that the point about the lactam and decan chain really gets to, I think, a somewhat different issue from epitope spread, and that is to the escalation of the immune response to other organ-specific or largely organ-specific antigens, because I really feel that the damage in autoimmune disease really comes when you get that escalation of the immune response. And I think it may be the influx of other cells to GAG or IA2 or
Yeah.
the zinc
Yeah.
molecule, whatever it is, that really are the signature that you're getting clinical damage, or clinically relevant damage.
I'm with you. That's what we were talking about. That is the issue of epitope spreading coming up. Yeah.
So your hypothesis puts the antigen-presenting cells central in this process. So you go back and look at some of the genetic data. Is there evidence for APC or dendritic cell unique markers that are associated with diabetes or with, yes?
Well, there are a number of unique markers associated with diabetes. Okay. How much are they APC specific and how much are they relevant in this whole process is where it's, as far as I know, not clear, not identified at this present moment. Yeah, but that's an important point. Okay, thank you very much.